All right. Hello, everyone. Welcome to this edition of MS Imaging Special Interest Group. Today, we have a special edition of our webinar in honor of the Halloween season, featuring the spookiest molecule in all of imaging, sugars. I'm Kelsey Bonadono, a graduate research assistant at the University of Tennessee, Knoxville, and I am joined by my co-chair, Jessica Moore, who will be facilitating the Q&A. Jessica, I think you're on mute.
Man, starting strong. Well, thank you all for joining us and for unmuting me. My name is Jessica Moore, and I'm a senior scientist in proteomics at Discovery Life Scientists. I'm also here to facilitate today's session. So to give you guys some background, today we're going to have two speakers, and we'll take all panelist-specific questions following each presentation. And so to ask a question, there's a Q&A box at the bottom of your screen. So please let those questions roll in, even as they're speaking. We'll save them all to the end of the talks.
Awesome. So we are going to begin our talk today by talking about sugar itself, glucose transporters, and the imaging of glucose. We are joined by Dr. Gus Gray, an associate professor in the Department of Physiology at the University of Auckland. Dr. Gray directs the Life Sciences Mass Spectrometry Research Platform and coordinates the University of Auckland Mass Spectrometry Hub. Dr. Gray has been recognized for his contributions in the field of ocular research, including the 2023 National Foundation for Eye Research Cataracts Research Award and recognition as a Sir Charles Herkes Health Research Fellow. His work has included the study of age-dependent changes in metabolic pathways within the lens, as well as the spatial distribution of small molecules and therapeutics within ocular tissue. Today, we are excited for Gus to share some of his work on glucose transporters, and with that, I will hand it over to Dr. Gray.
Thank you both for the very kind introduction. Just to allow me to share my screen. It's a real pleasure to be invited to speak to the group today. Okay. Can you see my screen in presentation mode?
Yes, it looks good.
Yeah, great. Okay. All right. Okay. Thanks again for the introduction. So today I'm going to be telling you a bit about some of our work in spatial metabolomics and proteomics to understand ocular lens transparency and cataract. So as many of you may know, cataract is a clouding of the normally transparent ocular lens, and it is by far the most prevalent form of preventable blindness in the world today. There are a number of risk factors for cataract, including aging, diabetes, and some lifestyle factors, including smoking, sun exposure, and some long-term drug administration. And while there is an effective treatment for cataract, which is a surgical intervention where the surgeon goes in and removes that opaque material and inserts a clear plastic lens, that treatment does come with a cost. And so some statistics, recent statistics out of the US suggest that if we were to try and tackle the current wait lists and the cataract surgery burden in the US, it would cost around $7 billion. However, one of the issues that we're facing is that we have an aging population and increasing prevalence of diabetes. So that cataract surgery burden is only set to grow, and those wait lists are set to increase. So in the lab here in Auckland, we are interested in developing non-surgical treatments to delay or prevent the onset of cataract. So I'm going to start today by showing you not a mass spectrometry image, but a confocal image, because I think this really beautifully describes both the structure and function of lens tissue, which, with the speed of a small child who's perhaps had a little bit too much Halloween candy, I'm going to try and describe to you today in the next few minutes. So you'll see in this micrograph the green cell membranes and the red gap junction proteins. And you can see in the top panel this really nice regular honeycomb array. And that structure lends itself to the smooth and uninterrupted transmission of light through the lens to hit the retina at the back of the eye to form your image. If we look at this in a cartoon form, on the left-hand side, you can see a schematic of the lens. And I wanted to draw your attention to a few important structural features. So it has an anterior epithelial cell monolayer, which is this yellow monolayer covering the anterior surface. And over our lifetime, cells in this particular region, the germinative region, divide, and they then migrate towards the lens equator, where they differentiate and elongate into these fiber cells, which are shown in blue. So we have this very highly ordered cellular architecture of the fiber cells out in the cortex. And as part of that differentiation process, the cells degrade all of their cellular organelles. That removes those organelles from the path of light, but it does place certain functional constraints, and I'm thinking particularly about metabolic constraints on those cells, particularly in the central part of the lens. So what we end up with is a cell age gradient. So the cells in the edge of the lens are very young. The cells in the center of your lens are as old as you are. And when we look at the lens in a spatial context, we often divide the lens up into this epithelial cell monolayer and then the fiber cells into these three main regions: outer cortex, inner cortex, and core. And I'm going to refer to those later on in the talk. Not only do we have a cell age gradient, we also have a protein and water concentration gradient, and that sets up our gradient of refractive index, which allows the lens to function as an effective optical element. So previously, we have applied mass spec imaging and at the protein level to look at the different distributions of proteins in the lens. So the first protein we've looked at is alpha A crystallin. This is a molecular chaperone. It's a structural protein. It's very abundant in the lens. And we know that with aging, it undergoes post-translational modification. So this is the intact, so the full-length alpha A crystallin at about 20 kilodaltons. You can see in a very young human lens, we've got the most abundant signal around the edge and then a lower abundance in the center. And as we age, the full-length alpha A crystalline is lost in the center of the lens. Where does it go? Well, I talked about post-translational modification. So we get extensive C-terminal truncation of that alpha A crystalline. So you can see a 1:101 residue form, most abundant in the young lens. But in these older lenses, it again is further truncated to these shorter forms, 1:58, 1:40, and many more forms. So this has implications for its chaperone function to mitigate any opacification and cataract formation, and also in lens optics in its contribution to forming that gradient of refractive index. So the other protein that we've looked at a while back in an intact mode is looking at aquaporin 0. So this is an integral membrane protein, relatively small at 26 kilodaltons, and also very abundant in the lens. And we think that it acts both as a water channel, but also as a cell adhesion molecule, but it also undergoes extensive post-translational modification. So here's an example of that, where in the cortex of the lens, we've got a prominent signal for the full-length aquaporin 0. And then in that coarse region, we've got truncation of the full-length aquaporin 0 to a shorter form truncated at residue 246. And here are those mass spec images just showing it's most intense in its intact form at the very edge, and that truncated form is more abundant in the very center of the lens. So that's some of the structural features that we can identify imaging proteins at the intact level. But I also mentioned about function of the lens. So the lens is avascular, doesn't have a blood supply. That removes that blood vasculature from the path of light so that we get even light transmission. But instead, the lens operates this microcirculation system, which experimentally measured externally has a flow of ions, water, and nutrients into the lens at its poles. and then outwards at the equator. And if we look at a more cellular level representation of that, we get ion flow. So the sodium potassium ATPases at the cell surface drive a current of circulating ions that travel in towards the lens extracellularly. That current, those ions are then taken up into the cells and then removed via an intracellular route mediated by those gap junctions, which I showed you in one of the first slides stained in red. This ion flow leads to water flow, and that water flow is mediated by the aquaporin 0 protein that I showed you. And then in addition to that water flow, you get convection of nutrients. So this is where we're really talking about our glucose and our glutathione as an antioxidant, which is a major protective mechanism for age-related nuclear cataract. So why am I telling you about this extensive and complicated lens function transport system? Well, the energy for driving this microcirculation is derived from glucose, of course. Glucose metabolism, formation of ATP, and then those sodium potassium ATPases utilizing that, that energy to support lens transparency. So when everything's working appropriately, we get normal vision, but if the structure or the function of the lens breaks down, of course, we get a cataract. So now we're talking about glucose and Halloween candy. So you obviously don't want to have too much blood glucose in your blood. And we all know that blood glucose is very well regulated under normal circumstances. But if you develop diabetes, or if you have diabetes from an early age, you are prone to the formation of what's called a diabetic cataract. This typically forms in that cortical region of the lens, leading to the disruption of that that organized cellular structure. And this is largely due to two different mechanisms. One, there's an osmotic stress, so the glucose is taken up into the lens and metabolized to sorbitol. Sorbitol is an impermeant osmolyte, so it draws water into the cells and you get this, the swelling and disruption of the cells. But there's also an oxidative stress component, so that glutathione is very important. Of course, mediating glucose uptake is solute carrier proteins. And in addition to the glucose transport, there's other solute carrier proteins that mediate the regulation of lens cell volume. So we have an interest in solute carrier proteins at the proteomics level. So with that introduction, what I want to step through today is first imaging glucose uptake and metabolism with mass spec imaging in a bovine lens. So that's one of the common animal models that we use in the lab. Then I want to show you how we're spatially resolving glucose transporters and their relative abundance, again, predominantly in our model system, and then a little bit of translational work. So looking at those SLC or glutes in multiple different species and highlighting one of the differences that we've found in human lenses. So for the first set of experiments, where's glucose transported into the normal lens? We're using a bovine lens model. We take lenses freshly collected from our local abattoir, so they're in relatively healthy state. We incubate them in an artificial aqueous humor that has a physiologically relevant concentration of isotopically labeled glucose. And we incubate for varying different times. We then take those lenses out, freeze them, section them, spray them with matrix. And here we've done FTICR-based multi-imaging to detect that exogenous glucose uptake and metabolism. So initial uptake looks to be in that germinative region and equatorial zone. So after 5 minutes, you see that signal appearing in that equatorial region. You wait a little bit longer, that signal increases before after about half an hour, you've got signal all the way around that outer cortex. We can do this multiple times, multiple biological replicates, and we can digitally dissect all of those spatial maps and show that, yes, indeed, what we're calling the equatorial region has that preferential uptake. And that makes sense because that's where those sodium potassium ATPases are concentrated. Not only can we look after a half an hour, but we can extend that time frame out to up to a day. And we can also then look at the metabolism of that labeled glucose. So here's just the parent molecule, the labeled glucose. Again, we've got that uptake in the germinative region. And then after about 16 to 20 hours, we've got signal pretty much throughout the lens, although interestingly enough, we're lacking the signal on the anterior surface. We then look at a direct metabolite of the glucose. This is glucose 6-phosphate. It's a similar pattern that we see, although the pattern is shifted in time, and that makes sense because the glucose 6-phosphate is a downstream metabolite of that parent labeled glucose. And then we can also look at the sorbitol formation. Again, remember, this is in a normal lens, so sorbitol is formed in a normal lens, but it is overly abundant in a diabetic lens. We can see its formation. This shows a slightly different pattern. We get collection or accumulation of the labeled sorbitol on the anterior surface first before being distributed throughout the lens. And we think that matches nicely with what we know about the enzyme activity of aldose reductase, which forms that sorbitol. So as an aside, before I get to the proteomics of this, I just wanted to share with you some of the data interpretation we've been doing in this space. So we've been able to collect up both biological replicates and all of those timed incubations of those bovine lens sections and overlay them in X and Y, attempting to put them onto a common X and Y grid. And then we're able to color code the timing of the appearance of those different signals. So we're sort of loosely calling these a kinetic multi-image. So earlier time points are in the red, later time points in the blue. And hopefully you can see for all of the different labeled metabolites that we see, we can sort of build up a timed map of their appearance in our model lens system. Then, of course, we can relatively crudely overlay that with the metabolic pathways that we know that are existing in the lens, and we're attempting to do this sort of comprehensive spatial mapping of glucose uptake and metabolism in a bovine lens. Of course, we can also do this in a diabetic lens model. We haven't yet done that, but that's certainly something that we're looking at. So after that slight digression, back to the main story. So we've looked at where glucose is taken up. Now the question is what transporters are mediating that uptake. So to do that, we've taken, again, our bovine lens and we've peeled off the anterior epithelium. So that's what is represented down in the left here. We've got an epithelial cell monolayer, and then we've just done a very basic micro punch manual dissection of those different epithelial cell populations, run gel-based proteomics, and we've found two different glucose transporters, GLUT1 and GLUT3. We've then done a relative ratio, an abundance ratio, and shown that GLUT3 is more abundant in the central epithelium and GLUT1 is more abundant in that peripheral epithelium and when we get into those fiber cells. And then we've come in with an antibody. Unfortunately, there's no really good GLUT3 antibody, but we've been able to label GLUT1 antibody. So on the top, you'll see cell membranes and cell nuclei labeled in red and blue, respectively. We've got our GLUT1 glucose transporter labeled in green in the middle panels. And you can see by panel I, which is in this region here, region 4, you can see the GLUT1 is abundant in those cells and it's extending into those fiber cells. And then we've been able to overlay a multi-image of that labeled glucose after half an hour, which seems to match nicely with the distributions of our GLUT1 transporter. So we think that GLUT1 being the most abundant transporter in those peripheral fiber cells, peripheral epithelial cells and the fiber cells, that is probably doing the most of the grunt work of mediating that uptake of glucose into the lens. So finally, we wanted to translate this to other species and look at the human condition in particular, because while there are some therapeutic eye drops that are somewhat effective in mitigating cataract formation in animals, the same is not true for humans. The same sort of approaches don't seem to work. So we're asking the question, what's different about the human lens? So we did, again, for our proteomics approach, we reverted to a relatively crude spatial approach. So we're just manually dissecting the lens. We collected up a whole bunch of different, like a farmyard of lenses, including human, microdissection, gel-based mass spec, swath MS, and then looking at those different relative abundances. So on the left-hand side, I've got the epithelial lens cells represented, and I just wanted to draw your attention to the abundance of amino acid transporters, which seems to be relatively well conserved across the different species. Mitochondrial proteins, which makes sense because those epithelial cells still have those mitochondria. And the glucose transporters here, so we can see relative consistency, and it's mostly made up of that SLC2A family, those glucose transporters. In the fiber cells, a little bit different. So the amino acid transporters tend to get turned down in most of the higher order species here. The rodents still maintain high levels of those amino acid transporters. Similar pattern with the mitochondrial proteins. And again, this makes sense because relatively speaking, those smaller lenses will have a higher proportion of mitochondria in the peripheral fiber cells before they're degraded to remove them from the path of light. But you can see a very consistent signal for our SLC2A, our GLUT transporters across the different species. So we focused, of course, on the GLUTs. We looked at GLUT1. We found GLUT1 in all different species. This is just a sequence coverage map. We found GLUT3 in most of the species. Interestingly enough, we didn't find it in the mouse and the rat. So there are a few subtle species to species differences. But importantly, we also found GLUT12, SLC2A12. So this was in a relatively young human lens. You can see the sequence coverage on the alpha fold representation here. And when we did the little manually dissected spatial analysis, we can see it's present in all different human lens regions, although it's a slightly different abundances. And then with the help of friends at Vanderbilt University with Professor Kevin Shai, we're able to do a study of its relative abundance with human lens aging. And so you can see in the outer cortex of all of the different ages of human lens, you can see the predominant form is GLUT1 in red. The second most abundant is GLUT12, and there's a little bit of GLUT3. So in actual fact, those relative abundances don't seem to change too much with age. They do change with position in the lens. So you can see decreasing amounts of GLUT12 relative to GLUT1 in the inner cortex and the core, and the disappearance of GLUT3 as well. So finally, we wanted to spatially localize GLUT12 in the human's lens. This is a novel result. So this is just the cell membranes in a human lens. This is GLUT1 in a human lens at the periphery. And as you would expect, it's membrane associated predominantly, although GLUT12 shows a slightly different pattern. So you can see it's mostly cytoplasmic in those very peripheral lens fiber cells. If we look in a different region, in that central core region, again, this is GLUT1. You can see it associated with the cell membranes. GLUT12 also associated with those membranes. So there seems to be a subcellular location switch for GLUT12, depending on whether you're looking at the cortical fiber cells or those core fiber cells. So the question is, what's it doing in the human lens? So we know from other tissue that there's two main things. One, it can function as a sodium-dependent glucose transporter. So it uses this energy in the sodium gradient to bring glucose into the cells. And two, in muscle or in some muscle, it's insulin sensitive. And so is there in actual fact a regulatable system for the uptake of glucose in a human lens that's sensitive to insulin? So these are questions that we want to answer going forward. So just to wrap up, what I've shown you is the multi-imaging results for the uptake and metabolism of labeled glucose in a bovine lens, and there's hot spots in that germinative region where the cells are growing, dividing, and migrating towards the equator. Gel-based proteomics to show the relative abundance of our GLUT transporters, and interestingly enough, we've got GLUT12 as a novel transporter in the human lens. So going forward, as I said, we want to look at modifications to GLUT12. We also want to look at its functional contribution to glucose uptake in a human lens and its regulation to understand whether it has a role both in normal lens function, but also in diabetic cataract formation. I'll wrap it up there. I really want to thank all of the people who've done all of the work, all of the funders that support our work over the years, and thank you very much for your attention.
Awesome. Thank you, Dr. Gray, for that excellent talk. We do have time for questions, so please remember to utilize the question and answer box at the bottom of the screen. With that being said, we do already have a few questions. The first one, kind of a double-sided question. What do you find the most useful to keep the spatial registry consistent for your overlay images is the first part of our question.
That's a really good question. So what I showed you was a relatively crude overlay where we were really just scaling the lenses in X to match a certain set dimension. What we're trying to do now is do a fancier version where we are tracing the periphery of every lens section, and then we are doing a non-linear warping of those sections to a standardized lens shape, and then we're overlaying them all on a standardized lens shape. So I've got a PhD student who's been working on this for a while. I would hate to say that it's simple. So he's been working hard on that, and we're hoping to have that slightly more advanced work out relatively recently. So that's all I can say on that, really.
Awesome. And the second part of their question, was keeping in the Halloween theme, is since you keep referencing Halloween candy, what's your favorite? What one would be worth a cataract for?
Well, okay, so I don't know whether you'll have these this candy in the US. I'm very keen on licorice all sorts. So they're kind of, you know, a little bit of licorice and then a little bit of brightly colored candy and a little bit of a stack, kind of a little sandwich. That's my go-to in New Zealand. If I were in the US, maybe Reese's.
My knowledge, I haven't heard of the licorice ones, but Reese's are a great choice. Another,
well, you can come to New Zealand and you can fill your boots.
I'll have to do that. Another question from Peggy Angel. So she said, Gus, great talk. Has anyone ever looked at proteases and lens aging?
Proteases?
Yes.
Yeah. So there's some work on proteases. We know that they are different in different species. So some of those truncations that we know occur to, say, aquaporin 0, they're at different sites in different species. And say in a rodent, it's driven by proteases. In a human, it's not driven by proteases. We think it's just driven by, in aquaporin 0's case, asparagine to aspartic acid, and then spontaneous backbone cleavage of the polypeptide. So we don't think there's a lot of specific protease activity in the lens. We think it's more of an age-related truncation.
Interesting. And a follow-up question that she had was, how do you see translating your findings to improve clinical care?
Good question. So I guess our ultimate goal is an eye drop with a compound that can get across the cornea and into the lens And for diabetic cataract, of course, it will be around, regulating glucose uptake potentially in the cortex. And that's a far more easily tractable problem because you're only trying to reach the cortex, that outer part of the lens. For an age-related cataract, you're trying to get to the very center of the lens. And so we've been looking at antioxidant strategies. So we've also been looking at combinations. So we need an effective molecule, whether it's in the diabetic cataract, a way to regulate glucose uptake or block glucose uptake or change its metabolism, or in the case of age-related cataract, it's an antioxidant. So we need the molecule, and then we are trying to utilize what we know of lens function and lens physiology to attempt to deliver those molecules to where they need to go. For example, that microcirculation system, we know that sort of declines with age. So if we could find a way to upregulate that in addition to delivering a therapeutic molecule, that might be a way to delay or prevent the onset of cataract. So kind of a dual type strategy, some significant scientific and also physical barriers to get past at the moment, but we like to think we're making some progress.
Very interesting. One last question, I think, before I hand this over to Jessica, was that glucose can be a tricky molecule to get into the gas phase. So how did you arrive at the NEDC matrix in negative mode? Do you have any tips for people who might be approaching similar small molecules for imaging?
Okay, so I certainly cannot, I'm not a chemist, so I certainly can't say it was with any great academic background that we chose that matrix. I think what was happening was we were already working with that matrix to look for antioxidants. So it's a great matrix for looking at glutathione, and somewhat by chance, we found a signal in that data that we thought was glucose, a chlorinated glucose, and it was also very prominent. So we did the work and decided that that signal in the lens at least was a mixture of myoinositol and glucose. So from there, we then went, okay, well, since we think we can see glucose, perhaps we can label it and track it. So that's what we did. So it was really sort of a by chance thing rather than a great chemistry-based strategy to identify appropriate matrices.
Awesome, thank you.
Yeah, I love when we kind of share stories like that within the community, because like sometimes you stumble upon like something that really drives your research. And so I love that. And so with that, mayhaps, we're going to thank you again, Dr. Gray, for the excellent talk. And I really like this. I like this webinar a lot, right? Because in the first part, we're going to focus on glucose, right? Glucose itself, glucose transporters, and the things that glucose can do in biology. And now we're going to transition to the second half of our session, where we're going to talk about what those sugars might be doing specifically to proteins. So please join me in welcoming Caroline Cottrell, who's an MD PhD candidate in the laboratory of Dr. Richard Drake in the Department of Pharmacology and Immunology at the Medical University of South Carolina. Caroline completed her undergraduate degree in biochemistry and molecular biology at Mercer in 2020. And she's interested in incorporating targets that are identified from mass spectrometry imaging into high throughput clinical assays, so translation, which could be used in diagnostic and prognostics. So during her time at MUSC, Caroline's generated some beautiful data sets and publications related to spatial atlasing of N-glycosylations within healthy and cancer tissues. So again, please join me in welcoming Caroline as she shares some of her work with us today.
Thank you, Jessica, so much. It's such a pleasure to be here. All right, so I'm going to go ahead and share my screen here. Get all the way to the beginning. All right, perfect. Everybody can see my screen. I'm going to go with yes. So like Jessica talked about, I'm really interested in spatial atlasing of cancerous and non-cancerous tissue. Specifically, I'm going to be talking today about pancreatic cancer and the evolution of the N-glycome and ECM proteome throughout the initiation, the development, and progression of pancreatic cancer, also known as PDAC. So just to outline the talk today a little bit, I'm going to start with some background information about pancreatic cancer and glycosylation and the ECM proteome and how all of those are related. Additionally, I'm going to address some of the relevant biological questions or gaps in knowledge our studies are attempting to answer. Also, I'll talk a little bit about the methods that we're using, specifically imaging mass spectrometry, MALDMSI, as well as show you guys some results from 3 different cohorts. So the first cohort is all primary pancreatic cancer tissue, so all from the first tumor that's localized to the pancreas. We also have some unmatched unmatched liver and lung metastases, as well as we have this really large TMA set. It's about 1,060 patients, and it spans the breadth of normal adjacent to pancreatitis, pre-malignant, as well as full-blown pancreatic cancer tissue. And all of these tissue resources are a result of a collaboration with the Washington University St. Louis, specifically the DiNardo lab, through their pancreatic spore. So first, to give a little bit of background of really what is pancreatic cancer. So pancreatic ductal adenocarcinoma, or PDAC, it accounts for 90% of pancreatic cancer cases. And it's really the malignant transformation of healthy exocrine tissue to invasive duct-like glandular structures. And these really take over the normal tissue architecture. So the cells that are making your digestive enzymes and also insulin, which as Gus mentioned, is a very important hormone for glucose uptake, those get obliterated. And so it can cause new onset diabetes, as well as digestive enzyme deficiency and malabsorption. But this is currently the 4th leading cause of cancer-related deaths globally. And you can see, as denoted by the darker blue, it's higher incidence rates in higher income countries like the US, Western Europe, some in Russia and Argentina, and a little bit in Australia. So most patients who are diagnosed with pancreatic cancer, it occurs in the 7th and 8th decades, but mostly in people who are older than 50 years of age. So why is pancreatic cancer such a deadly malignancy? So there are multiple factors that play into this. One of them is that many lifestyle behaviors, such as obesity, are on the rise, and alcohol use that can contribute to pancreatic cancer incidence. Additionally, there's really no screening assays that are specific or sensitive enough to diagnose pancreatic cancer in the general public. I'll talk about one that's used clinically really for assessing the recurrence of pancreatic cancer and the response to chemotherapy, but none to apply to the general public. So patients, since there are no screening assays available, it's usually pancreatic cancer is diagnosed at an advanced stage when the disease has already metastasized, and it makes curative surgery an impossibility. And surgery in itself is already difficult because the pancreas has such intimate relationships with the vasculature within the abdomen. So the majority of patients, when they're diagnosed, they have advanced metastatic disease, systemic disease, so they need systemic therapy. And clinicians right now are really broadly using cytotoxic chemotherapy cocktails such as FOLFIRINOX. And there are very few effective targeted therapies. There are some mutational status specific KRAS inhibitors that are out right now, but those kind of apply to a minority of patients. Additionally, there are some KRAS and pan-RAS inhibitors being approved clinically right now. As far as immunotherapy goes, it's really proved revolutionary in the treatment of hematologic malignancies and some other solid malignancies, but for pancreatic cancer, there are very few patients who respond. So how do glycans and pancreatic cancer, how do they kind of interact? Kind of before I get to that, I'll talk a little bit about N-glycosylation and the glycocalyx. So you can see that the glycocalyx here in this transmission EM on the center of an endothelial cell, and it's all these little hairy-like projections. And those are really the first things that each of your cells see about one another. It's the communicative interface for all cells. And it's made up of a lot of N-glycans, O-glycans, proteoglycans, syndicans, a lot of different cans, if you will. And those are constantly being shed and turned over in patient serum as those cells die. And one of those glycans that is a glycoantigen that's shed is called CA19-9 or Cialis A. And I've shown it here. It can be attached to N glycans, O glycans as well. And it's shed in patient serum, and it's one of the glycoantigens that clinicians use today to really track how a patient is doing as far as responsive to chemotherapy or even surgery. To get more specific into N-glycan structure, so N-glycans are these oligosaccharide chains that are attached to proteins via an asparagine residue, and they're one of the most common post-translational modifications. Over 80% of proteins have them, and these are three classes: the high mannose, multi-antennary, and bisecting N-glycans that make up kind of the glyco world. So I've kind of talked a little bit about how glycans can be used as biomarkers for serum assays, but what about their functional importance? What are they really doing to promote cancer? So N-glycans can modify different proteins within adherence junctions, such as E-cadherin, and really affect the way that cells bind one another and allow them to release from those binds and travel to other places to enhance migration, as well as it can modify different integrins and discoidin domains to promote the binding to different ECM peptides. So then now these cells can traverse through the ECM and travel to other places. Additionally, different glycans can modify receptors such as receptor tyrosine kinases like EGFR to promote cancer cell proliferation. And they can also modify other receptors like VEGF to enhance angiogenesis. Finally, glycans can bind, specifically I'm talking about Cialis A and X glycan moieties can bind lectins within our endothelium as far as for within vasculature, and can promote metastasis of cancer cells. So what about the ECM in pancreatic cancer? So this is just an H&E of a healthy pancreas, and you can see how cellular it is. So we have all of these acinar cells. Those are the cells that are the exocrine cells that are making all of our digestive enzymes. And here's our islet of Langerhans that are producing insulin. And when we have cancer within the pancreas, you see how decellularized it is. It's all this pink stroma. It's desmoplastic stroma is abundant. And we actually have within a tumor, very little of the tumor is made up of the actual adenocarcinoma or the cancer cells. It's majorly the ECM proteins or desmoplasia. And so generally, the ECM is known to serve as a physical barrier for the transport of drugs, as well as for nutrients and oxygen for immune cells to kind of fight the cancer. But these ECM peptides also have signaling events of their own as far as enhancing in many of the same ways that glycans kind of enhance cancer progression. And I should know also that many of these ECM proteins, all of them have N-glycosylation sites. I think an average of 4 N-glycosylation sites per ECM protein. But as I was saying, all of these ECM peptides, they can cause different signaling events that enhance cell proliferation, migration, as well as can cause a differentiation of immune cells from tumor suppressing to tumor promoting phenotypes. And here in the bottom left corner, I'm just showing the superstructure of a fibrillar collagen and showing the triple helical domain that's typical of these, as well as we're going to be looking specifically at the peptide level, so the peptide sequence here. So now to get at a couple of gaps in knowledge that we're attempting to address, really, we want to know, can we dissect the different cellular neighborhoods within the PDAC tumor microenvironment using N-glycans or ECM peptides? So do tumor cells have specific ECM peptides or N-glycans that they express versus non-tumor cells? And when we look at the metastases, the liver and lung metastases, do their N-glycomes or ECM proteomes look very similar to the primary pancreatic cancer, or do they look more like their native tissue of lung or liver? And then kind of looking more broadly, more translationally, can we correlate specific N-glycome profiles or ECM proteome profiles with clinical outcomes such as survival, treatment response, disease aggression, et cetera? So the way that we're addressing these questions is through an imaging mass spectrometry approach, specifically multi-MSI. And all the tissues that we have are formalin-fixed paraffin-embedded tissues. that go through some initial processing, dewaxing, rehydration, antigen retrieval. And then we apply these two different enzymes, pingase F, to cleave off our glycans, and collagenase 3 to cleave all of our ECM peptides that have these triple helical domains. We then immobilize our N-glycans or ECM peptides with our CHCA matrix, so they stay in their in situ location. This matrix also allows for ionization via our MALDI-TOF. And as Gus kind of talked about, we have this laser that rasters across all of our tissues. And so you get these heat maps that show the distribution of our different analytes, with the more red being more abundant, with more blue being less abundant. And specifically for our ECM peptides, I'm not really showing our proteomics methods here, but we did a traditional scrape proteomics with tissue homogenization, followed by LCM SMS. But we also have some liquid surface extraction methods we're using now. So to talk a little bit about the primaries, so we have this cohort of 20 primaries, and I'm showing the H&E stains here with the areas of tumor outlined in red. And you can see that the dark purple cells, that are actually the cancer cells, are really the minority of cells within the tumor microenvironment. It's mostly that desmoplastic stroma. We also have our lymph node in green and our immune cell clusters circled in black. And these are 3 N-glycans that are a part of a larger class of N-glycans called bisecting N-glycans. So you see the presence of this square here. These are N-acetylglucosamines, and their presence denotes a bisecting N-glycan. We also have all of these are core fucosylated here, as well as they have outer arm fucosylation. And so we saw that kind of visually that these were typical of our areas of cancer. Additionally, so all of these pancreatic tumors have multiplex IHC, which was performed by 2 students in Dr. David DiNardo's lab, Blake Sells, an MSTP student, and Alice Cow, a PhD student. And what we were able to do is they stained specifically for CK19, which is upregulated in pancreatic cancer cells. And Emily Butka, who is a computational biologist, was able to overlay the multiplex IHC staining with our areas of tumor, and found that these 2 N-glycans shown here were expressed majorly in the tumor cells and not as much in our non-tumor areas. So next, to talk a little bit about the ECM proteome. So like I said, we did some LCM SMS proteomics, and Jade McDonald assisted me a lot with this. She is now, she's graduated from Dr. Angel's lab and is now at Vanderbilt with Dr. Spraggins. And when we did our proteomics, we found a little bit over 4,000 different unique peptides, and we were able to match back 121 of these to MALDI MSI peaks. And when we looked at kind of the different types of ECM peptides that were detected, we found that majority of them were fibrillar collagens, and that really lines up with what we see in literature as far as these fibrillar collagens are most abundantly secreted by cancer-associated fibroblasts within the PDAC tumor microenvironment. So this is just to show you a few representative images. Again, we have 3 primary tumors with our areas of tumor outlined in red. And we can see the kind of the abundant colocalization within the tumor microenvironment of these two different collagen peptides with the stromal areas of our cancer. And these are both COL1A1 peptides. And you'll see this peptide specifically here, 1343.67, come back a few times. So next, I wanted to show you some results from our liver and lung metastases. So shown here, this is a liver metastasis that's circled in red. It's the tumor, and the lung metastases circled in red here. And we find that the same N-glycans that were detected in our primary PDAC tumors are also detected in our liver and lung. More so, we also have spatial transcriptomics data on this specific liver met. And we were able to segment the liver met into 3 different cell populations of tumor, immune cell aggregates, and stroma. And we repeated the analysis using Pearson correlation coefficients to see, okay, what N-glycans are specifically colocalizing with this area of tumor. And we found the same N-glycans that colocalize statistically with the tumor here in our met also co-localized statistically with our tumors from PDAC. And just to show you some clustering, so we took ROIs from the normal adjacent liver, normal adjacent lung, the primary PDAC tumor, as well as the liver met itself and the lung met itself. And what we found is by the N-glycome, the PDAC, the liver and lung met all look very similar, clustered together according to their N-glycome and away from their normal adjacent tissues. And you can see this in this PLSDA as well. So looking at the ECM proteome, it's a little bit of a different story. These ECM peptides do look specific for areas of tumor, but they also have some signal in areas of normal tissue as well. And we see that with our hierarchical clustering, that really the PDAC clusters away from the normal adjacent liver and lung, which clusters most closely with the liver and lung metastases, showing that the normal adjacent liver and lung and the liver and lung metastases look more like each other or more similar than the PDAC. Really, this is what we're thinking and theorizing, is that it's more so, rather than the PDAC bringing its own ECM machinery to these different tissues, really the PDAC is co-opting the ECM machinery that's already available within the adjacent liver and lung. So next, these are some initial results from our TMA analysis. And something I also kind of want to emphasize here as well is that this analysis was performed on a slide-by-slide basis rather than a core-by-core basis, just to get some initial trends. Each of these slides was homogeneous as far as pathology, pancreatic cancer versus normal adjacent versus premalignant versus pancreatitis, but just to give you an idea of the general trends of our N-glycome. And we see that we really have two distinct groups of pancreatic cancer. And again, this is based on a slide-by-slide basis. So there may be some differences within the individual cores that we're going to flesh out with this, such as maybe some of these patients were treated with neoadjuvant chemotherapy, or they had better survival outcomes, those types of things. When we look at this volcano plot, and I made this basically by categorizing, comparing all of the PDAC N-glycome versus everything else. So the PDAC N-glycome versus the N-glycome of pancreatitis, premalignancy, and normal. And what we found is, again, these same N-glycans that we've been seeing throughout pancreatic cancer, throughout the primary pancreatic cancer, through the metastatic pancreatic cancer, still pop up. We still see these bisecting N-glycans with core fucosylation and also some outer arm fucosylation. And you can see that the expression of these N-glycans is very heterogeneous. So it's not throughout all the cores. It really is on a kind of a core by core basis. So it'll be interesting to see what the differences are on an individual patient level between each of these cores. So now looking at the ECM proteome, you can see all of our PDAC clustering together, and we see that with increased disease severity, so more PDAC, we get higher expression levels, relative expression levels of these different ECM peptides. We also have this one group of pancreatitis that's at the higher end of our ECM expression. So we think something that it kind of makes sense within the biology of pancreatic cancer is that one of the precursors for pancreatic cancer can be long-standing chronic pancreatitis, which does produce a lot of desmoplastic stroma also. So it'll be interesting to look back at the H&Es to see, is this also as extremely desmoplastic pancreatitis, and that's why it's pairing with the pancreatic cancer. Looking at our volcano plot and looking specifically at ECM peptides that are upregulated in cancer versus non-cancer, we see this COL1A1 peptide 1426 pop out as being upregulated, and then this COL4A1 peptide 858 is being downregulated in our cancer. And when we look at the representative images, they tell a similar story of, we see this COL4A1 peptide 858.399 is overexpressed in our normal and not so much throughout the rest of the disease states. And we think that makes sense biologically because the COL4A1 peptides are part of a family of collagens that make up basement membranes. And a part of the development of cancer is really the invasion and the destruction of basement membranes to allow for the development of carcinoma. So it really makes sense that we're seeing it only in our normal areas. Additionally, we're seeing the abundance of this 1426, which I showed in the volcano plot, as well as this 1343 collagen peptides, these COL1A1 peptides, they're increased in our PDAC, but you can see they're also increased in pancreatitis and pre-malignant cores. And what we're thinking is that that's showing that we're setting up the niche within the ECM to allow for malignancy to set in way before malignancy is actually present. So to just go over a kind of a few conclusions, a little bit of a summary is we really found that these bisecting N glycans and fibrillar collagen peptides are typical of primary pancreatic cancer, and that we can compare the N glycome of primary tumors with unmatched liver and lung metastases, and they look very similar. Additionally, it looks as though from our ECM proteome results that the METs appear to be the result of adapting native tissue ECM machinery, as well as some preliminary results from our tissue microarray establishes the N glycome of primary tumors are similar across statistically powered patient cohorts, and really show distinct expression profiles when you're comparing the disease states such as normal, adjacent, pancreatitis, and pre-malignancy. Additionally, it really looks like the ECM proteome sets up a niche for malignancy even decades before the development of cancer. Something that we're doing next, as I kind of alluded to earlier, was that we're going to do a core by core analysis to really look at the individual patient characteristics and how they may be differentiating the N glycome and ECM proteome. So with that, I really would love to thank the Mad Lab team. We're absolutely a collaborative group, and I would not be successful without that. So thank you to the Drake Lab, Angel Lab, and Meta Lab, as well as the DiNardo Lab at Washington, St. Louis. My funding through the training grant, the TL1, as well as the patients who donated their tissues. Thank you so much.
Awesome. That was a fantastic talk. So thank you for that. And then we're happy to take audience questions. So it's the little box on the bottom. And please do ask questions, because if not, I'm going to ask you some maybe hard ones. So you got the trick of the trick or treat.
Yes, I did.
So I love that in your bio, you said you're very passionate about non-invasive biomarker monitoring. Me too, right? That's something that I really like. And I like this idea of this glycocalyx. That's a word I learned from your talk. And so I was wondering, as you see some of these reordering in like the extracellular matrix, something that's very tissue specific, how you think we can translate these to a non-invasive biomarker. And I was thinking, as you talked about this glycocalyx in terms of like things like exosomes, things that shed, and how that might alter some of your sample prep or some of your thoughts on that.
No, absolutely. Thinking about kind of different ways to assess these things. So kind of like I talked about, glycans are constantly shed, as well as these ECM peptides are shed into our blood. So definitely one way I can envision it is, of course, serum. Getting patient-matched serum, especially in earlier stages of pancreatic cancer, is always a challenge. And that's something that we see too. Right now, we do have some patient-matched serum for our pancreatic cancer, but it would be really great to get it at the pre-malignant stage, right? Some other ways that we can kind of look at it in a non-invasive manner, a lot of these patients go through EUS, which is a type of endoscopy, I should say bronchoscopy, where they basically sample the cyst fluid or the pancreatic fluid. That could be another kind of source for these different biomarkers for sure. Specifically for looking at different exosomes, I'm not exactly sure about how that would differentiate sample prep, but I think you're absolutely right. It's something that I think we've talked about a lot in prostate cancer specifically in our lab, but not so much in pancreatic.
Yeah, that's great. So one of the fundamental things that you guys did in this research is that you use mass matching, right? Mass matching from MALDI to mass matching to ESI. And I was wondering if you'd comment on maybe the risks of that, and then, some of the strengths.
That's definitely something. So right now, I feel like that's really the part of my PhD that I'm diving into currently. We just got a new LCMS system, an EvoSep. So I'm kind of divulging that right now. But no, you're absolutely right. As far as I feel like getting the mass matches, I already see kind of issues with it as far as like drift. So processing the samples in batches and over time can absolutely cause some of those differences. I see that even in kind of our MALDI data. I can see some of the drifts. So like applying the appropriate window for each of those peaks to make sure that we're capturing them. It is more difficult with peptides because the MDZ values are so much closer than for glycans. So that's definitely a challenge for sure.
All right, we have one more. Are you able to determine if any visualized N glycans are directly from the glycocalyc shedding? And then how does glycocalyc shedding impact cancer progression?
So I think okay. So one thing that we're doing right now is glycoproteomics. So we're really trying to look at the proteins that these glycans belong to really give more of a biological basis to what we're seeing as far as we can look at, on the enzyme level. So MGAT3 is the enzyme that the glycosyltransferase that's responsible for making these bisecting and glycans. But what receptors or proteins are these glycans modifying? That kind of goes back to getting more at the biological significance of it. But one class of N-glycans, I mentioned high mannose N-glycans, are very typical of proliferative cells. And so we see it in highly metabolic tissue. So I could see that those are related specifically maybe more to the glycocalyc shedding. But it would be interesting to see if we could correlate some glycans specifically with, more cell death or apoptotic or necrotic proteins to see if they're related.
Yeah, that'd be a great tool for spatialomics. Last question, maybe, we'll see. Do you have any ways to study which glycans are associated with the ECM peptides?
So yeah, we can do it in those same ways as far as with the glycoproteomics to look at, can we see that these specific ECM peptides that are modified by these specific N glycans. We can also do it kind of in a quote unquote dirty-ish way with looking at overlays as far as with our ECM peptide profiles and our N glycans and co-localizing those. But I would think that glycoproteomics would probably be the best.
Awesome. Well, wonderful. This was a very great talk. And so, in the last little bit of time, I want to thank everybody for joining us today and both of our excellent speakers for today's MS Imaging Special Interest Group featuring sugar and just in time for Halloween. So, thank you all again for joining us. And once again, thank you to our speakers.